Welcome back. You've probably heard at least one reference to the so-called central dogma of biology before, the idea that DNA leads to RNA, which ultimately leads to the production of proteins. This set of concepts is essential to biology, and of course, it's also going to be essential to your biochemistry section on your MCAT. In this lesson, we're going to take a look at some of the ways you might have to explore the central dogma of biology on test day using a real-world application. In this lesson, we're going to take a look at the ways that a mutation can actually lead to a problematic protein, as well as a few different molecular techniques that can be used to analyze this process. Let's get started. Take a few moments to read over the given scenario. You may already be familiar with the disease progeria. It has striking, unfortunate physical effects on affected children. Werner syndrome is a form of progeria that we're told affects adults. And interestingly, the disease is caused by a mutation to a single protein. There are many genetic diseases that involve the malfunction of a single protein, and in most of them, there are several different variations of a mutation to that protein within the affected disease population. Doesn't always have to be the same mutation. It just has to cause the same ultimate effect to cause the same disease. In this case, that cause is a non-functional WRN protein. Our question stem researchers know this, and we're told that they run several tests to better characterize what kind of mutation may be the problem and where it could possibly have occurred in the DNA. In this session, we're going to think about the clues the researchers uncovered and try to decipher from these clues what the underlying mutation could be. We were already told that WRN protein is a helicase and it's used in replication repair machinery. However, without that information, what would you have expected a helicase to do? You'd probably expect any helicase to unwind double-stranded DNA in preparation for incoming DNA replication machinery. A typical helicase would be involved specifically in synthesis. And as it turns out, WRN helicase isn't so different either. We're told it's involved in single and double-strand break repairs. Given that it's a helicase, we could hypothesize that it still functions to unwind DNA, but for a different reason. For repair, quite possibly post-synthesis. This also tells us something else potentially really important about WRN helicase. If you were to run an assay in a cell looking for WRN protein, where would you expect to find it? It's still a helicase, and it still interacts with DNA, so it must be found within the nucleus. Now that we know where to find WRN protein in a healthy person and what we would expect it to typically be doing, let's start exploring the specific mutation affecting this subpopulation of Werner's patients. The first test described was a northern blot, which we're told gave data that there was a WRN mRNA present in the correct quantity, but at a lower than usual weight. What kind of mutation is implicated by a change in molecular weight in an mRNA transcript?
If the weight of the transcript is too low, there must be some portion of the transcript missing. Our most obvious guess is a deletion mutation. Ultimately, this could be a mutation that affects how the heteronuclear RNA was spliced, creating a truncated mRNA strand. In either case, our ultimate mRNA strand has been substantially shortened from its expected length.
In order to figure out where that deletion may fall, let's take a look at a representative strand of the mRNA. Starting from the outside and moving inwards, what is the purpose of the 5' cap and the 3' tail? Both of these regions are added on post-transcriptionally, and they function to protect the mRNA transcript. Without these regions, the mRNA would degrade far more quickly than normal within the cytosol. Adjacent to the cap and tail are typically untranslated regions, one at each of the ends of the mRNA transcript. If these regions are never to be translated, why do they exist as part of the mRNA? These regions do a lot. They help to facilitate translation, localization, and stability of the mRNA transcript over time, making them essential components of the overall transcript. The final region remaining is the coding region of the mRNA, the portion that codes for the actual protein. However, this region doesn't just code for a protein's function. In certain specialized proteins, like WRN helicase, the coding strand also must include the localization signal. For proteins working in the nucleus, this is referred to as the NLS, or nuclear localization sequence, and it must be present to ensure the protein can enter the nucleus. Remember, the nucleus has to be protected from many foreign infectious agents, things like viruses and bacteria, which is why this specific sequence is so necessary, even though it's often really small. Now take a moment and consider all these possible regions of the mRNA. What part of this molecule can you be sure was not affected by the mutation to the DNA? The mutation could not have been to the cap and tail, as they are added after transcription occurs, during post-transcriptional processing. Now that we've recognized that it's a deletion mutation and eliminated a couple of possibilities for the cause of the overarching problem, let's deal with our next piece of evidence. The protein that these WRN patients are making does actually work. Let's start with the big question. If the protein itself does function, what do you know can't be part of the problem? The portion of the coding sequence that actually codes for the functioning protein can't be the site of the mutation. This may seem a bit counterintuitive, but remember, there are many ways to mutate DNA in such a way that mRNA is affected, and those mutations do not have to be to the protein function coding sequence in order to cause problems. We know this particular WRN mutation is causing problems for a fact. If the protein's function isn't the problem in these patients, what else must be wrong with their protein that would cause disease symptoms in patients? If the protein is working when it's present, the problem must be that there just isn't enough of it.
This fits with our remaining possibilities for mutated regions. If the UTRs or the localization sequence is the site of the problem, the mRNA stability or the protein's location would be negatively impacted, and it wouldn't be able to function adequately in this patient population.
Let's take a look at some data to see if this hypothesis about concentration is corroborated. Take a look at the Western blot image, either on the screen or in your lesson book. As you can see, this image displays quantities of protein within different sample cells in different cell regions. What does the Western blot tell you about this population of WRN patients? This figure demonstrates that, compared to a normal nucleus, there's substantially less WRN protein present in the nucleus of these Werner's patients. That absolutely fits with our earlier hypothesis. Concentration is definitely the problem. The researchers in this Western blot analyzing protein concentration have confirmed that the functional WRN protein is in insufficient concentrations compared to normal controls. Now, let's take a moment and explore what you might do next if you were the researcher. You've got two possibilities, a mutation to a UTR, which would affect overall stability in translation, or a mutation to a nuclear localization sequence. If you could measure additional protein concentration data and compare it to this data, what would you want to look at to tell the difference between these two types of mutation and identify the specific problem? I know this is a tough application question, so let's talk all the way through it. If the mutation were to the UTRs, we definitely expect to see this lower protein concentration in the nucleus within the Western, because we would expect the transcript to have a tough time ever being translated. In fact, you would expect to see lower protein concentration everywhere if the mutation were to a UTR. On the other hand, if the mutation was just to the localization sequence, that thing that lets the protein get into the nucleus, there would be low protein concentration in the nucleus, but normal or even high levels of WRN protein in the rest of the cell. The best way to tell the difference between these two mutations would be to compare this data to an additional measure, WRN protein concentration for the whole cell. If the overall concentration is too low, it's a UTR mutation. If the overall concentration is normal, you know it's a localization sequence mutation. This was a really tough question, so if you managed to predict that solution, phenomenal job. Now let's bring this problem to a close by remembering just what it was this mutation actually did. In the normal central dogma of DNA leads to the synthesis of RNA, which is used to code for the proteins that help run almost all major functions in every single cell in our body. It's pretty mind-boggling to think about. When a mutation occurs, problems at any level of this process can affect the entirety of a normal central dogma function. In other words, a problem with the DNA can lead to a potential problem with a protein.
The original mutation was made to the parent DNA strand, possibly a deletion or a change to splicing sites.
As a result of this mutation, what happened to the mRNA and the protein produced by the WRN gene? As a result of this mutation, the mRNA transcript was shorter than it should be, and this led to a protein that wasn't able to consistently exist in sufficient concentrations in the nucleus. Now that we know what exactly has happened in these patients, we can analyze their functional outcome. Given WRN helicase's normal function from the original scenario, what would happen in a cell that didn't have enough WRN protein in its nucleus? Without those repair functions, the nuclear DNA will build up strand break mutations and damage over time. These same issues are seen in the DNA of many cancerous cells and in the DNA of some cells that have degraded telomeres. These cells and their nuclei will functionally age at a far faster than normal pace, resulting in many symptoms, including, in this case, the quite literal appearance of rapid aging. Though there's no current cure for Werner's patients, this type of logic, figuring out the cause of a disease, is the first step in developing something to ultimately treat even the most devastating of illnesses. As such, this is hugely important in life, and that's why it's going to be such a huge part of your training as a physician and your MCAT experience. In this lesson, you saw quite a few different biochemical techniques in action, as well as the various ways that a mutation could lead to different problems with a protein. Going forward, the central dogma is going to continue to be one of the most testable aspects within the biochem section of the test. You could see anything from easy problems to tough real-world application problems or experimental passages on your MCAT. Make sure to be prepared by practicing this content regularly right up until test day.